So before we're going into the actual experiments that you were supposed to do in this lab, let me back up a little bit. We got cut off slightly at the end of the first recording here for this lab. So in the end, it kind of got fast. Let's just back up from here. Polymer versus monomer. So these macromolecules, these food molecules that you're eating, a lot of them are polymers. And if you're eating a fat, it's composed of, fat would be considered a polymer of glycerol and fatty acids. So these two are the components. And if you're eating protein, and proteins are composed of amino acids. So the protein is the polymer and amino acid is the monomers. A starch, that's a polysaccharide, and that is a polymer of simple sugars. So those would be such as glucose, monosaccharides. So glucose or monosaccharides are the monomers, and the starch would be the polymer. Of course, your body is doing all of these wonderful enzyme reactions to break down these polymers into monomers for one purpose, and that is we want to absorb these nutrients. They need to first be absorbed into the blood and then delivered to the cells that need them as building blocks, either for energy or to make your own proteins. And absorption is extensively covered in the gastrointestinal lecture. That will be more a lecture kind of topic. And the pH, the last term here, we definitely got cut off on that. So that's the logarithm of the hydrogen ion concentration. That was definitely covered in your chemistry lecture. The pH is important. So on a simple level, the pH is a scale that goes over 14 orders of magnitude. It is a logarithmic scale, so that means every change in the number of the pH, so if, let's say we're going from 7 to 8, is a 10-fold change in the hydrogen ion concentration. So you're spanning quite a few orders of magnitude, 14 to be exact. The point of neutrality is 7, and anything less than 7 indicates a higher hydrogen ion concentration. That means more acidity, and the negative logarithm to be exact of the hydrogen ion concentration is getting smaller. So that means if we have a pH of 2, we have quite a high hydrogen ion concentration. I'm sure this was explained in lecture, so I'm going to do this here briefly. If you just want to use the pH scale, really what you need to know is that it's a scale that goes from 0 to 14. The point of neutrality is 7. Anything higher than 7 is a base, and that means the hydrogen ion concentration is getting increasingly smaller. And if the numbers are less than 7, then we have an increasingly higher hydrogen ion concentration. We increase the acidity. In the human body, the most acidic place would be the stomach, and it's estimated that the stomach acidity is about a pH of 2 to 3, somewhere around that, which is the pH that pepsin really loves. Now, before we actually go into the experiments, let me do one more thing, and that is the indicator reactions. You do need to know the indicator reactions, because how would you know that amylase did its job in digesting starches and releasing simple sugars into a solution if you have no way to detect starch versus simple sugars? So let's make a box with the indicator reactions. Okay, so here it is, the indicator reactions that you need to know about. Starch plus iodine is something that you already know. It makes the solution turn dark blue. Simple sugars can be tested for with Benedict's solution. Now, and do know that last time in the last lab you got to use these heuristics. So I showed you the heuristics. Of course, that's an easy way to check for simple sugars, but in the lab we usually use a sky blue solution called Benedict's solution. And if simple sugars are present and you're heating up the solution, then you get a rusty red precipitation. The heat is an absolute requirement. If you don't heat up the solution, no reaction will occur. But Benedict's plus simple sugars, it's a simple lab test. It's pretty sensitive to. If you're heating up the solution, then if you see a rusty red kind of, at first, if the concentrations are low, it's kind of orangey. But if the concentrations of simple sugars are higher, it turns into a rusty red precipitation. And that indicates that simple sugars were present. Another indicator reaction you need to know is the pH change. We're using litmus in this lab. Litmus is a reagent that is kind of blue when the pH is above 7 in solution. We're using it to digest evaporated milk, which has a high fat content. And so in the fat digestion, the release of fatty acids, you follow that with the pH change. And the litmus reagent, it makes the evaporated milk look kind of a very light lavender, kind of pastel, bluish, purplish. And if the fatty acids are released, then the color will change to this more pastel pink. It's not a very dramatic color change, but if the pH drops below 7, which it does when fatty acids are released, then you're going to have this pastel pink kind of color. Okay, so now let's take a look at the experiments that you would be doing if we were on site and you got to do these experiments. Here is some more write-up, introduction, all the stuff that I just explained with the terms. You can read through that, kind of read up on some of the terms if you need more explanations. And so this is all explanatory. And here is the succinic acid reaction to fumaric acid. That's a redox reaction because the succinic acid is the substrate, of course, and then the enzyme is succinic acid dehydrogenase. And we do this experiment with hamburger meat because it has a lot of meat muscle tissue. And so it contains the succinic acid dehydrogenase already because muscle tissue, as you might know, has a lot of mitochondria, and this reaction occurs in the mitochondria. And so therefore, if we just buy the hamburger meat, fresh hamburger meat works really good. You have lots of mitochondria in this muscle tissue, and it does this reaction all the time. So it will then oxidize the succinic acid to fumaric acid, which is the product. So fumaric acid, after oxidation, succinic acid becomes fumaric acid. And the hydrogens that are being taken off of the succinic acid, they are being parked onto FAD. And you can see that right here. So then the FAD gets reduced to FADH2. The competitive inhibitor to succinic acid is malonic acid. It looks very much alike to succinic acid, but it does not produce the product, the fumaric acid. So it's a false substrate. That's what we call it, false substrate. And here are your instructions. Of course, you don't get to do this, which is kind of sad, but sort of you have to do this all theoretically. Kind of think about it. Let me just walk you through this. So there's a bunch of experiments in this lab, and the first thing you're going to try out is the controls, which some of them, at least I showed you in the last lab already, that the detection of starch with iodine. So that's one of the controls right here. And so there'll be iodine. Iodine is used to detect the presence of starch. So we follow a starch digestion by first staining the starch with iodine, so it's dark blue, and when the enzyme amylase and digests the starch away, then the starch is gone and it's just simple sugars, and then the blue goes away with it. And now you need to detect the presence of simple sugars, and that is done with Benedict solution. So here, these are sort of your indicator control reactions, and I'll show you what that looks like. I have some pictures, some images in the next recording that I'm going to show you. Experiment B is a pretty elaborate experiment. The environmental effects on the enzymatic hydrolysis of starch. So basically you're testing the enzyme amylase in different conditions. You vary the concentration, you vary the temperature, and you change the pH and kind of test out under which conditions this enzyme works the best. And so it involves quite a number of test tubes, and you, as you can see here, you're changing the amount of amylase in the test tube, you're changing the pH in the test tube, so, and then you're also varying the temperature in the end. We'll take a look at the results, but the way it's set up, you can follow that. The emulsification of fat is a very simple experiment. You can try that at home and kind of visually observe that. If you have like a skinny, like a test tube type of piece of glass, or maybe what would work is a skinny glass, or maybe like a shot glass type of thing, but something transparent. What you could do is fill it about halfway with water or 2/3 with water, and then add a couple drops of salad oil, like olive oil or salad oil, whatever you have in the kitchen. You can add that, and then use your hand, the palm of your hand, to seal the top and then shake it really hard. And what you will see is that the whole the solution turns cloudy. And the reason for that is that with the mechanical shaking, you are breaking down the fat into some very small droplets, and at first they will stay in the water dispersed, but then because fat and water, they don't like to mix, the fat droplets, they will fuse back together, and then eventually you have your salad oil back on top, and then the water on the bottom, and everything is transparent again. Now, what this experiment does, we'll set up one test tube just like that with water and a couple drops of salad oil, and another test tube, same thing, plus you're adding also the bile salts. And the bile salts work as an emulsifier, and so what it does, it will prevent these fat droplets that you've generated. It prevents them from fusing so fast, and so it keeps them smaller longer. That's exactly what emulsifier does. Also, you can maybe, if you want to try that experiment, or you may have already done that experiment, leaving your sunscreen or some sort of a lotion in the car when it's really hot in summer. And sometimes that causes the emulsifier to fail, and then your lotion will break apart into the oil or the fat component and the water component, and it doesn't look pretty. So you can try that too if you like. Use a small amount of like a sunscreen or something, put it in a small bottle, leave it out in the heat, and watch the emulsifier fail, and then you have separation into oil and water again. Okay, that's experiment C, and experiment D is the digestion of fat by pancreatic juice. For that we use evaporated milk, which has about 30% fat, and then we're adding the pancreatic juice, which contains lipase, and the lipase will then digest the fat into glycerol and fatty acids, and the fatty acids, once they're released, they will change the pH, and we follow that pH change with litmus reagent. And so as soon as the fatty acids are being released and they're changing the pH, then the lavender blue from the litmus reagent, it will turn into a very light, like a pastel type of pink, but you can see it. It's enough pH change where you can see it. So we're starting out with making both test tubes slightly alkaline, so above 7, so the litmus is blue, and then the test tube that has the pancreatin solution, so the lipase is contained in that pancreatic juice. That one will change color and will turn kind of pinkish. I'll show you results for that. The protein digestion, there we're testing the enzyme pepsin in different conditions, and the way this experiment is run, we're using photographic film, and you might think, or X-ray film pieces, you might think, oh, how does that work? Well, X-ray film is basically sort of in black and white photography. You're using like a plastic backing, and it's coated with a gelatin material that's embedded with silver granules, and then if they get exposed to light, they turn dark. But anyway, so we're using undeveloped film, and the gelatin is protein, of course, and it's sitting on top of the X-ray, this plastic backing, and once we are using pepsin, if the pepsin works, then it will digest this gelatin coating of this plastic backing. It will digest it away, and you're left with a clear piece of plastic if the pepsin worked. So I'll show you results for that too. In experiment F, you're just going to simply use about 10 milliliters of non-fat milk in this case, and we're going to add a small spoonful of renin to that, mix that up, and watch it turn into a semi-solid kind of consistency within about a half an hour, 45 minutes. You will see that the milk turns into more of yogurt consistency, so the renin will separate the milk into the curds in the whey. The last experiment looks at the succinic acid dehydrogenase activity, and for that we're using hamburger meat. It's like a hamburger meat shake, basically, that contains enzyme and all the ingredients that we would need to run a Krebs cycle, because it's meat, muscle, that is. And so we're filling about 2 centimeters worth of this hamburger shake into the test tubes, except for the control number 4 test tube, and then adding these ingredients. You succinic acid, here malonic acid, the inhibitor, and here this one just gets water. And then we're adding methylene blue to each test tube, so they're all blue. And as the succinic acid is being oxidized, actually the hydrogens will be transferred to methylene blue and change the color of methyl blue to colorless.